Okay, welcome back to the second half of chapter 15. We're still focusing on lipids. Now to refresh your memory, lipids are broken up into two different categories. The lipids that contain fatty acids and then lipids that contain the steroid nucleus. Out of the ones that contain a fatty acid that we've talked about so far are the waxes, which are just an ester of one fatty acid and one alcohol. Okay, and then we talked about triacylglycerols, which are lipids that have that glycerol backbone and three fatty acids. Okay, glycerophospholipids are broken up into whether they're either fats or oils. Okay, and that's where we stopped with part one. In part two, we're going to talk about glycerophospholipids and steroids. So here, this is just a refresher slide. So this is the triacylglycerol that we talked about in the last lecture, where we have our glycerol backbone and the three fatty acids. A glycerophospholipid, which we're about to talk about, is very similar to this. The only difference is down here on the very bottom fatty acid. We're going to replace it with a different group. So glycerophospholipids are the most abundant lipid in a cell membrane. So that's going to be important. I'm going to talk about some other things that make up a cell membrane, but I do want you to know glycerophospholipids are the most abundant biomolecule in the cell membrane. They're composed of glycerol, two fatty acids, a phosphate group, and then an amino alcohol. Okay, be able to identify that. Glycerophospholipids have two nonpolar fatty acid chains. So remember, those two fatty acids that are on the top are considered nonpolar. And then the phosphate group with that amino acid is a polar group. These are three of the, sorry, I said amino acids, I meant amino alcohols. These are three of the amino alcohols which end up being bonded to that phosphate group. I don't expect you to memorize these. I do want you to know they're polar though. Okay, that's important. So here's a picture of a phospholipid. Glycerophospholipids can be shortened to just being called phospholipids. Here's a picture of one. Notice that the phosphate group with that amino alcohol is polar. And remember, polar and nonpolar things don't mix. So what tends to happen is, since this carbon-carbon single bond here can freely rotate, the polar side ends up rotating to the opposite side of the two nonpolar fatty acid groups. So the fatty acids are nonpolar and the phosphate amino alcohol is polar. I want you to know that. And there's your glycerol backbone. So the structure and polarity of a glycerophospholipid. So I just talked about this before. What I want you to see though is right here, we often symbolize a glycerophospholipid as just having a polar end with two tails. If you remember the soap molecule, the soap molecule had one polar head and one nonpolar tail. So this is kind of how we symbolize a soap molecule. This nonpolar tail embeds itself in the grease, and the polar head stays outside by the water, and that's what makes the micelle. But when we symbolize a glycerophospholipid, we often just do this as the polar head, and that encompasses the phosphate group and the alcohol. And then we have two nonpolar tails. So that's kind of the quick way to draw a glycerophospholipid. Focusing on cell membranes, there's a lot here I want you to know. Okay, your biology teacher is going to expect you to come with a certain amount of knowledge about the cell membrane. So cell membranes separate the cellular content, the stuff inside the cell, from the external environment of the cell. It consists of a lipid bilayer. So lipids are the number one thing that make up your cell membrane, and to be more specific, it is the glycerophospholipid bilayer. It's made up of two rows of phospholipids, okay, or glycerophospholipids. It has an inner portion made of the nonpolar tails from the glycerophospholipid, with the polar heads on the outside and inside surface of the cell. So I'm going to back to this slide. I just kind of want to bring this point home. When we were talking about the soap molecule, we had our blob of grease, if you remember this. And the soap molecule, the polar head stayed out the outside and the grease embedded itself onto the inside. Remember that? That's what made the micelle. But that's different for soap, because soap we had grease, which was nonpolar on the inside, and it had water, which was polar on the outside. So we just needed a single layer of soap molecules to be that interface between the inside of the micelle and the outside of the micelle. The thing that's tricky about the inside of a cell is that the inside of the cell is polar, unlike your grease. So we have a polar region on the inside of the cell, and then we have a polar region on the outside of the cell. So in order to deal with that, you need two layers of glycerophospholipids, because you're going to have the polar head, which contains the phosphate group and that amino alcohol, on the outside of your cell, and the nonpolar group on the inside. Well, since the inside of the cell is polar, you're also going to have another polar head, and the nonpolar tails are going to want to hang out together. So you end up needing a bilayer to separate the polar inside of the cell from the polar outside of the cell. So the lipid bilayer not only contains mainly glycerophospholipids, but it also contains some protein, carbohydrates, and cholesterol. The two fatty acids in the glycerophospholipids used for cell membranes are unsaturated. And that makes the cell membrane fluid-like rather than rigid. Remember fluid rather than rigid. Your cell walls are not just, even though we tend to think of them as kind of this spherical shape, they're actually very flexible. And that's due to the carbon-carbon double bonds that cause the kinks in those fatty acids. So since there's some unsaturation in that fatty acid, those glycerophospholipids can't pack real tightly together when they form that cell membrane. The lipid bilayer contains protein, carbohydrates on the surface that communicate with hormones and neurotransmitters. Here is a picture. There's a lot on here that I'm not going to expect you to know. I'm going to go over it here real quick, and then I'll kind of simplify what I expect you to know. So here's our cell. If we focus down here in the bottom right hand corner, this is the nucleus of the cell. This is the cytoplasm, which is the polar liquid intercellular fluid. And then we have our cell membrane, which separates the intercellular fluid from the extracellular fluid. And so if we look at what they're doing is they're pulling out a small portion of the membrane. And let's kind of go over some of these things. So the plasma membrane is another word for the cell membrane here. It has some glycoproteins and some carbohydrate side chains, which we talked about. It has a glycolipids is another portion. The glycerophosphobilayer, so that's just saying that's what this whole region is. It has a hydrophobic region. Now remember, hydrophobic means water-fearing, and that is the nonpolar tails. So the hydrophobic region is the nonpolar tails of the glycerophospholipid. The hydrophilic region then are the polar heads, which are that phosphate group and that amino alcohol group. And then we said there was some cholesterol. The polar heads, the hydrophobic region, are the full polar areas, and the hydrophobic region is nonpolar. So it's important to know that on the inside of the lipid bilayer, right in here, is the nonpolar region, or the hydrophobic region. On the outside of the cell and on the inside of the cell is the polar region, the polar heads. Okay, so that you have to read that carefully about, are they talking about the inside of the cell, or are they talking about the inside of the bilayer? Okay, the inside of the cell is polar, but the inside of the cell membrane, that bilayer, is the nonpolar region. So what do I expect you to know? There's a lot on here. I just kind of mainly go from the previous slide. There's carbohydrates, there's proteins, there's cholesterol in the cell membrane, but the most abundant molecule of the cell membrane are the glycerophospholipids. Here's another picture showing you kind of the same thing but from a different angle. So once again, there's a lot more detail on here than I want you to know. I'm going to save that for your biology teacher. So to simplify, what I expect you to know is I want you to know the cell membrane is composed of two layers of glycerophospholipids. The inside of so let's go over this here. You don't need to know this part. The glycerophospholipid bilayer So I do want you to know there's 2 layers, and it's made of phospholipids, also called glycerophospholipids. I do want you to know it has some proteins. It has some cholesterol in there. It doesn't look like it's showing any carbohydrates, but I do want you to know that. And this is important. The hydrophobic tails are on the inside of the bilayer. And are they showing us the polar heads? The hydrophilic heads down here. They're on the inside of the cell, and we also have some of them out here on the outside of the cell. That kind of sums up what I expect you to know from the lipid bilayer. Okay, switching gears. So we've already covered now the lipids that contain fatty acids. So now we're going to move on to the lipids that contain the steroid nucleus. So these steroids are going to be your cholesterol and your steroid hormone, steroid hormones. Focusing on what the nucleus, that steroid nucleus, looks like. It's got 3 rings that are made of 6 carbons and 1 ring that has 5 carbons. Notice there are no fatty acids in the steroid nucleus. Focusing on cholesterol, it's the most abundant steroid in the body. It has some methyl groups. So here's some methyl groups. It has one real big alkyl group. And it has the steroid nucleus, and it has this hydroxyl group on it. If I was to ask you which part of cholesterol is found probably on the inside of the cell membrane, and which part of cholesterol do you think is on the outside of the cell membrane, I would hope you would say that this whole region right here, since it just contains carbon and hydrogens. Remember, I've said several times in this class, hydrocarbons are always nonpolar. So if it just contains carbon and hydrogens, you want to look at that and say, okay, this is a nonpolar region. The OH group, remember, that's very polar because it can hydrogen bond. So this OH group is the polar region of cholesterol. So if I asked you what part of cholesterol would be embedded inside the lipid bilayer, I would hope you would say this region over here that's nonpolar, because the lipid bilayer, the inside of the lipid bilayer is your nonpolar hydrophobic region. Cholesterol in the body, it's obtained from foods such as meats, milk, and egg. It's synthesized in the liver, so not only do we sometimes eat it with what we eat, intake it in our body, but our body actually synthesizes cholesterol in the liver. And we need cholesterol for things like the cell membrane. We need it for our brains, nerve tissue. It is the precursor for steroid hormones and vitamin D. So our body actually makes vitamin D by starting off with cholesterol. Cholesterol is known for clogging arteries when you get high levels, which will form the plaque. So this is really tied to that LDL levels of the hormone. Levels of cholesterol are known for clogging the arteries. Over here we have a picture. Kind of cool. This first one here is a normal open artery, so you can see it's all open. But here we have an artery that's clogged with cholesterol plaque. So what would I want you to know from this slide? I do want you to know that cholesterol is synthesized in the liver. I think we all have a good idea of what things we eat, but that would be pretty much it. Cholesterol in food, so this is kind of fun just to look at. Cholesterol is considered elevated in the blood when it reaches 200 milligrams per liter of blood. It's synthesized in the liver and obtained from the food. So those are the 2 sources of cholesterol. So let's just kind of look at some of these for fun. Liver here, if you have a 3-ounce slice of liver, you can be getting 370 milligrams of cholesterol. Eggs are high in cholesterol. Lobster's up there. Here's fried chicken. Hamburger. Here's chicken if you take off the skin, so not so bad. If you look down here, margarine has no cholesterol. So margarine has a bad name, remember, because of the whole partially hydrogenated. So remember, we get margarine from hydrogenating oils. And what that does, the concern with margarine is you have those trans fatty acids that occur during the partial hydrogenation process. But one of the good things about margarine is that if you're worried about your cholesterol, is that there's no cholesterol in margarine because it came from oil. However, you do have to worry about the trans fatty acids when you're having have in margarine. So I'm not going to ask you any test questions from this slide. It's just kind of fun to look at the differences in our food. Okay, so remember, cholesterol is a steroid. Hormones are, many of the hormones are also steroid hormones. So some of them here to look at. Steroid hormones are chemical messengers in our cells. Some of them are sex hormones like androgens in males and estrogen in females. And we also have some steroid hormones that are the adrenocortical hormones, which are synthesized in the adrenal glands. Two examples of those are the aldosterone, which regulates electrolyte and water balance in the kidneys, and cortisone, which increases blood glucose levels, stimulates synthesis of glycogen in the liver. So those are some examples. I'm not going to ask you anything really this detailed. I just want you to know that steroids are important for cholesterol and hormones. Looking at some of these just for fun, I won't ask you any test questions on this. But steroid hormones are produced from cholesterol. They include the sex hormones such as androgens, also known as testosterone in males, estrogens, also known as estradiol in females. Here's the structural formulas of them. So this here is testosterone, which its biological effects is it plays a role in the development of male organs, male sexual characteristics including muscles and facial hair, sperm formation. Here's the structural formula of estrogen in females. It's produced in the ovaries. It plays a role in the development of female sexual characteristics, plays a role in ovulation. Here is a formula of progesterone, which is also produced in the ovaries. It plays a role in preparing the uterus for the fertilized egg. Here's a look at some adrenal corticosteroids. So here is the structure of cortisone right here. It increases blood glucose level. We talked about this one already. The aldosterone increases the reabsorption of sodium in the kidneys, retention of water. Prednisone here has a big name in the pharmaceutical companies now, is known for reducing inflammation, treatment of asthma and rheumatoid arthritis. So those are just some examples I put in just so you could kind of see them. Notice the steroid nucleus. That's the main thing I'd want you to be able to do, is if I showed you one of these molecules, I wouldn't ask you which one, is cortisone or anything like that. But I would want you to look at them and say, oh, this is a steroid. And I want you to know steroids are lipids. Okay. Another grouping of steroids are the anabolic steroids. These are the ones we're hearing about in the news with the athletes taking steroids to buff up. So anabolic steroids are derivatives of testosterone used illegally to increase muscle mass. They have side effects including fluid retention, hair growth, sleep disturbances, and liver damage. So here's a few of them. I wouldn't ask you any of these specifics. And this is a fun slide to kind of sum up the lipid chapter. Kind of cute. How did, so this real skinny, scrawny football player, how did you bulk up so much this summer? This big guy says, substance abuse, frankly. No, not steroids. No, Ben & Jerry's Heath Bar Crunch. So, Heath Bar Crunch having our lipids that are fats and oils, things like that. Steroids can also bulk you up.